There it is. It's right here.
Hello, thank you very much for coming to this talk. The green plants encompass important diversifications along evolution because they have generated a number of organisms that have populated the oceans and freshwater environments, and also they have given rise to terrestrial ecosystems. We are interested in this study in documenting the timing of green plant evolution and also trying to identify major radiations through their evolution. In this study, we include a representation of all major lineages of green plants, including the charophytes, a sample of streptophyte algal lineages, as well as all of the lineages within the land plants. And we combine molecular information of extant organisms to provide phylogeny with information of fossils to calibrate the tree and to estimate divergence times. In this presentation, I will be talking about the methods and the results for the phylogenetic analyses, the divergence time estimation, and the diversification analysis. The taxonomic sample includes 192 species, including 4 red algae, which were indicated as outgroups, 45 chlorophytes, 7 algal streptophytes, and a number of representatives of all of the lineages within the land plants, including an increased representation regarding previous studies of the moss, of the bryophyte lineages, and of the ferns. The data set includes more than 17,000 base pairs coming from 2 nuclear markers and 7 plastid markers, which were aligned with FIDE, and we conducted a maximum likelihood analysis with RAXML. We conducted 5 independent analyses, each with 100 bootstrap replicates, and we selected from them the result that had the overall highest likelihood value, although the results were very, very similar. This is the obtained phylogeny. All of the branches are highly supported except cases like here or here, in which you can see thinner branches that had a weaker support. The chlorophytes form a clade, which is the sister group to the streptophytes, which also form a clade. And there is a basal grade of algal streptophyte lineages, in which Cara is the sister taxon to the embryophytes, and the Signamatales are the sister taxon to Cara plus the embryophytes. These relationships are very weakly supported, and in some of the maximum likelihood trees, the position of Carales and the Signamatales was inverted. And this was the only topological difference that we could find among our different maximum likelihood analyses. Within the land plants, there is a bryophyte grade at the base in which the deepest split within the embryophytes separates the liverworts. Then the second deepest split separates the mosses, and the hornworts appear as sister to the tracheophytes. These relationships are strongly supported and remain constant, and appear similar to some of the recent genomic analyses that have been done. Within the embryophytes, the lycophytes are sister taxon to the rest of the tracheophytes, and within the ferns or monilophytes, the deepest split separates a clade that includes Xylotum and the Ophioglossales from a clade in which Maratiales are the sister taxon to the leptosporangiate ferns, and Equisetum is the sister taxon to Maratiales plus the leptosporangiates, although this clade is very weakly supported. Within the gymnosperms, within the seed plants, in the gymnosperms, the cycads are the sister taxon to a clade that includes ginkgo, plus conifers, including the gnetophytes. And in this case, the gnetophytes appear as the sister taxon to cupressophytes, although this relationship is weakly supported. And within angiosperms, the branches along the spine of the angiosperms are very short. So I did this little cartoon where you can see that Amborella appeared as the sister taxon to all other angiosperms, but this other clade that excludes Amborella is very weakly supported. Then Austrobaileales are the sister taxon to Mesangiospermi, and within Mesangiospermi, the monocots are sister to all of the other ones, and Chloranthales is sister to Magnoliates, and Ceratophyllales is sister to the Eudicots. So we used this phylogram to estimate divergence times with BEAST 1.8. These analyses turned out to needed to be pretty intensive. We ran 500 million generations. We achieved convergence, but for a few of the estimated parameters, we could not achieve an effective sample size of 200. And to conduct these analyses, which here I am showing in a cladogram, we used several types of calibrations. We used 3 different types of calibrations that were introduced as uniform priors. The first is a uniform prior around the split between the red algae and the green plant lineage. And this uniform calibration between 1,623 and 1,393 million years old was obtained from an analysis conducted by Parfrey et al. So this is an calibration, a secondary calibration. We also implemented a calibration as a uniform prior around the embryophyte crown node. In this case, the lower limit was derived from the oldest fossils that can be assigned to the embryophyte lineage, which correspond to spore colony-coated cryptospores. And the upper limit of this uniform distribution was derived from plant fragments that contain spores that very possibly belong to liverworts and therefore represent a limit for the crown group of embryophytes. We also used uniform prior to calibrate the angiosperm crown node. This uniform prior went from 151 to 132 approximately, and it's derived from the estimate of the age of angiosperms obtained by Silvestro and collaborators in a recent study where they estimated ages using a quantitative study that considered the full fossil record of vascular plants. In addition to these calibrations, we implemented 38 additional effective calibrations as minimum ages, which were introduced as log normal priors all across the green plant lineage. This is the estimated tree. We could see that some of the divergences were very ancient. I will just be mentioning a few of them. For example, the split between chlorophytes and streptophytes is dated back to the Mesoproterozoic. The crown group of the embryophytes was dated between 485 and 473 million years old. So this is very important because this age indicates the onset of the diversification of plants on land and of the formation of terrestrial environments. The origin of tracheophytes, of vascular plants, is dated between 467 and 438 million years old. And this signifies the origin of larger plants, with three-dimensional branching systems that actually formed forests that at this stage occupied places very close to bodies of water. But these were forest environments. The split between monilophytes and spermatophytes is dated between 436 and 405 million years. And this is very important because in addition to indicating the origin of the ferns, of the lineage of the ferns, it indicates the origin of the so-called lignophytes, which are the stem lineage of the spermatophytes, which is the crown group. And in this lineage, in addition to the origin of seeds, which is very, very important, we also have the origin of wood in a very strict sense. In other words, wood formed by a bifacial cambia. So these plants were very large trees that, because they were very large trees and had seeds or seed precursors, were able to form woody forest communities detached from bodies of water. So this date signifies the onset of something like the conifer forests that we know today. And then the angiosperms are dated as being between 152 and 149 million years old. And this also, of course, indicates the origin of plants that eventually gave rise to terrestrial ecosystems as we know them today. We used this dated tree to conduct a diversification analysis using BAM 2.5, and this is a method that implements a compound Poisson process to identify shifts in the rate of diversification among phylogenetic branches and through time along each of these phylogenetic branches, which is the difference in colors that you can see here. And it does so by doing a reversible jump Markov chain Monte Carlo that produces a posterior distribution of shift configurations. And each shift configuration represents a particular combination of the number of shifts and their position on the tree. We conducted the BAM analyses, obtaining species richness for clades derived from the plant list. Our analyses had 100 million steps, and as a prior of the expected number of shifts, we implemented a number of 10 as recommended in the BAM manual. To identify which are the significant shifts from the non-significant shifts, we considered the marginal odds ratio of a shift occurring on each particular branch. In this tree, the length of the branch is proportional to the marginal odds ratio of a shift occurring on that branch. In some cases, maybe for example here, it shows that you cannot associate a shift to a particular branch, but more to a phylogenetic region. To show the shifts with the highest marginal odds ratio that we found, I will be using a so-called phylorate plot in which the colors represent the average rate of diversification estimated considering all the shift configurations found in the posterior distribution. So the oldest shift that we found corresponds to a clade that includes the 4 chlorophytes, although in this case I believe that the position of this shift might be too basal because it was not possible to distribute finely the diversity in the clade among the lineages because that information was complicated to obtain. So therefore, it's possible that what we are seeing here represents a trickle-down effect from one or more radiations that occurred higher within this clade. We found a shift in diversification not associated to the embryophytes, but to the clade that includes the Sigma Michaelis and Aurelis. There is also a shift within the liverworts associated to the younger Manidae, which correspond to the derived leafy liverworts. And within the mosses, there is a shift in the rate of diversification around the origin of the Dicranidae and Rhyidae clade, or it could be in a branch lower, which also includes the Phanaridae. Within the ferns, there is an increase in the rate of diversification somewhere around the origin of the leptosporangiate ferns. It could be exactly associated to the leptosporangiates as a whole, or to a branch further up, which excludes the Osmondalis, or even a branch further up, which also excludes the Hymenophyllales. And also within ferns, there is a shift in the rate of diversification associated either to the Polypodiales or to the Cyathiales and plus the Polypodiales. There are several shifts within angiosperms. These are distributed around the mesangiospermi. It could be either in the mesangiospermi or in one branch below or one branch above it. And there's also a shift within a clade within the monocots that includes all of the monocots except the Acroales and the Elismatales. And also, there is a shift around the origin of the pentapetalae, either in the petapetalae or including Gonorales and including Coxales and Gonaria. Finally, This shading represents the origin of the shifts within the angiosperms, and we can see that while some shifts seem to be correlated, or at least rather they seem to be contemporaneous, specifically the one associated to polypodiales, others within the land plants seem to be much younger. So to conclude, I think we're presenting a comprehensive estimation of divergence times among green plants, and we identified some of the major radiations. However, I think that in order to better understand which are the factors that might be associated to these increases in diversification, and also to see if there are correlations between diversifications by one clade in another clade of plants or with other groups of organisms, it's extremely important to include a much denser taxonomic sample. And I would just like to finish by thanking my co-authors, Dietmar, Quandt, Krug, Louise, and Francois Lutzoni, as well as a student and collaborator who helped in very particular parts of this research. And if there's time, I can take questions.
So Susana has agreed to answer questions for 15 minutes.
Great talk. I was just curious, because it was last year, Jeremy Belden and colleagues had a paper looking back at the 2010 paper, where they were discussing, they got very old ages of Triassic angiosperms in the 2010 paper. They could have revisited that and suggested that heterogeneity, molecular rates and weights would actually be responsible for